In this video, I am going to talk about the structure of ATP. So ATP is made up of three components. First is the nitrogenous base, which in the case of ATP is an adenine. So adenine is a double ringed structure. This sixth member ring of adenine, which has two nitrogen, it is a pyrimidine ring, and this five membered ring, which is also containing two nitrogen, is called an imidazole ring. So these pyrimidines are present this pyrimidine is present in the purines, and this imidazole is present in amino acids such as histidine. So the other component of ATP is this ribose sugar. This is a five membered ring, and it have five carbons. At the fifth carbon of this ribose sugar, phosphate molecules are added, and three phosphate are added, so that's why this molecule is called adenosine triphosphate. These phosphates of ATP are negatively charged, so there is a crowding of negative charges here. In this diagram, you can see that hydrogens are added to the oxygen, but in the cell these hydrogens are not added to this molecule, and this oxygen has negative charge. So due to this negative charge, this ATP molecule is very unstable, and it is also a reason for the high energy that is released when a phosphate group is removed from this ATP. This phosphate of ATP is the alpha phosphate, this phosphate is the beta phosphate, and this terminal phosphate is the gamma phosphate. 7.3 kilocalories of energy is released when this gamma phosphate is removed, and the bond between these phosphate is called phosphoanhydride bond. Here is a diagram showing the ATP hydrolysis and ATP synthesis. ATP is hydrolyzed into ADP and inorganic phosphate, and energy is released, which is 7.3 kilocalories per mole, and it is an energy releasing process. The ATP synthesis is an energy requiring process in which ADP and inorganic phosphate are combined and ATP molecule is produced. I already talked about that the phosphates of the ATP are negatively charged. So there is a force of repulsion between these negative charges, which makes this molecule very unstable. Inside the cell, positively charged magnesium ions are present. So this positive magnesium binds to these negative charges and reduces the repulsive forces between these charges. So it makes this molecule a little bit more stable. Another important thing related to this charge stabilization is that when a nucleophilic species, nucleophile is a species which has extra electrons and generally have a negative charge. When that nucleophile tries to attack this phosphoanhydride bond, these charges make it very difficult for this species to attack these phosphoanhydride bonds and cleave the terminal phosphate. But when magnesium is bound to the ATP, it reduces the negative charges, it reduces the repulsive forces between these charges, and it makes it a favorable molecule to be attacked by a nucleophile. And when a nucleophile attacks this molecule and cleave this terminal phosphate, energy is released, and that energy is used by cell for cellular processes. Thanks for watching.